Hello everyone, welcome to my YouTube channel. So here today we are going to be looking at our endocytosis and our exocytosis. How is the mechanism? So endocytosis is the active uptake of substances into a cell by the infolding of the cell membrane. So infolding, we are simply talking about how it was opened like that, then it folds inwards to close like that. So this is what we refer to as our endocytosis. So this is the definition. So it is the active uptake of substances from the outside by the cell via the infolding of the cell membrane. So the cell membrane will fold inwards. Like for example, it would look like something when something is trying to enter like that. The cell membrane is looking like this, but it will change its shape to look like something like this. So this is the infolding that we are referring to, the infolding of the membrane. So the three types of endocytosis is what we are going to be referring to: phagocytosis, pinocytosis, and the receptor-mediated endocytosis. So when we look at our phagocytosis, which is more like food substances as well as bacteria are swallowed like that. So let's try to create an animation. So let's imagine this animation. So what we should think of is we are now dealing with our phagocytosis. It's only that there are a lot of diagrams here, so that's why I have gone on where the pinocytosis is. But think of this as having the phagocytosis there. So what we are going to have is that, let's say this is a bacteria or a food substance, then here the cell membrane is closed. Then what begins to happen is that as the food goes like that, you will find that the cell membrane begins to open up. When it opens up, it is able to trap the food substances or bacteria like that. Then it will be able to close up. After it closes up, it is able to form what we are going to call the food vacuole if we are talking about the phagocytosis, where it has to do with the food. So as we can see, it is enclosed like that. Then this is taken into the cell. Pinocytosis, on the other hand, is the one that we are now describing. This is where the cell intakes substances such as liquids and those substances that are able to dissolve. So let's imagine that these are substances that are able to dissolve and they are having some liquid parts from the outside. Then what begins to happen is that it moves like that, it moves like that, it moves like that. When it is closer to the cell, it's not that the cell is just stationary. The cell will also be moving, but as it is moving, it will be able to, more like when it sees that there is something nearby, it will be able to open up like that. When it opens up, then it is going to more like swallow those substances. When it swallows those substances, then it is going to close up like that. Then it is going to make a vesicle. A vesicle is more like a transport protein. So let me just clarify that. So a vesicle is made of our lipids. They are transport sacs that are made of lipids. So these now will be able to trap those dissolved substances as well as the liquids. Then they will be able in the cell like that. For the third type of endocytosis, this is the receptor-mediated endocytosis, whereby substances that only binds to the receptor. Now remember, receptors are specific. So just as I have said, under this one, these substances that bind to the receptor like that are the ones that are enveloped or enclosed. Then we have another one there, it comes to this receptor like that. When they bind, then the cell membrane will be able to more like envelop or rather close up there. Then what begins to happen is that this now will break off from the cell membrane and it will begin to move like that. Here it is. Then from here it will be taken into the cell. So now endocytosis is very, very important in the sense that it allows certain substances to be able to enter the cell that cannot just enter via the cell membrane. Like substances such as white blood cells and hormones can be able to enter into the cell via this mechanism. The other importance is that substances such as food from the outside can also be taken in that can be used by the plant or animals. So depending on where this is happening, plants or animals can use this mechanism. Exocytosis, on the other hand, is the process by which the plant or the animals export certain substances. Export, we are simply saying that it takes them out of the cell like that. And also other components such as waste products. So this is how it's done. So it is enveloped. When the vacuole envelops some food substances or waste products or signaling substances, they are taken out of the cell like this. So this outer layer is more like the vesicle, which is the whole thing that has some substances inside. So as it begins to move like that, it is more like it will begin to join what we are going to call the cell membrane. It will integrate itself in the cell membrane like that. Then it will be able to open outwards. Then it will be able to more like export some things out of the cell like that. So this is exocytosis. So the vesicles are mostly made from the Golgi body. Then they are transported out like that. So this is very, very important in transporting certain substances that will form, let's say, signaling on the cell like that. Some glycoproteins and some glycolipids are transported in such a manner. And also the cell will need to export or rather remove toxic substances, and exocytosis plays a very crucial role. Let us just compare. So before we compare, we have this structure like this. So it's moving like that, the vesicle, then it will join the cell membrane, then it will release certain substance. So as we can see, this is the newly formed vesicle like that. Then it fuses itself with the cell membrane, then it releases wastes outside the cell. It does not always have to be waste. It could be useful substances such as proteins that will form on the surface of the cell membrane. So the difference is that endocytosis, the substances will be moving inside. Exocytosis, the substances will be moving outside like that. So that is our main differences. Thank you very much for watching. Hope you would like, subscribe, comment, and share for more. See you in the next tutorial.